Dog was just disappointed when I had to leave, as you might guess. All right, so I'll pick up where we left off. Let's see. All right. So this is we're looking really at microbial activity in a physiological sense at this point, and and really the only thing that to me was relevant, and we get to it later, is this when we start talking about parasitic activity and pathogens, and particularly the idea of what we're going to see a lot of with the obligate parasites, but that's coming in the future, so it's a good introduction to see that basically a lot of these organisms are unable, some can function just fine in the environment by themselves, and some require basically hijacking us, whether it's cellularly or intracellularly on surfaces for their nutritional needs. So, I mean, maybe that's the take home point from this. And all of these textbooks do the same thing. Where do I don't care where you find hydrogen. I know where you get oxygen, that type of stuff, nitrogen, don't care. Don't care, don't care, don't care. Okay. Well, so we start to get into this, and really, this is not, to me, the salient features of this particular chapter are the various classifications based on oxygen and temperature and water and acid and base requirements, and particularly the bacterial growth curve. To me, that's really the important part of what we're doing here. So my estimate is we will probably finish this, maybe not likely today, but probably Monday. And then, so I'm thinking maybe like a week. I usually like to do a week after class. So maybe whatever that is, at the 8th, something like that. Is that the Wednesday? I think. I think isn't the first, yeah, isn't that the way it's 31st is a, yeah, right? Right? Yeah. I figured that's the last day of the transfer window for the Premier League. So you never can tell who's being acquired. It's not football, it's football with a U. Wonderful game on today. The FA Cup, Man City and Arsenal. Oh, no soccer devotees. Maybe the two best teams in the world right now. Okay. Nevertheless, transport. This is a review. This is just movement across membranes and how things bring things in and out. Cytoplasm, even in organisms with cell walls, of course. When we start talking about, again, most of the time the driving force continues to be what it's been for our cells, concentration gradient. So we know what diffusion is. Molecules are constantly moving. Where they're higher concentrated, they collide, and they go to areas where they're less likely to collide. And we know what osmosis is. And osmosis is probably more relevant in microbial aspects of things, as I've commented on before, because microbes really require water. Nearly so when with the exception of the endospore, once that has changed, at that point the absence of water is devastating to their ability to function, to reproduce, etc. So, and again, selectively or differentially permeable mechanisms. Typically, water moves in very easily. Other things do not. Okay. And then we begin to look at, and this is the whole idea, the whole nugget about osmosis. Whenever we have differing concentrations of solvent to solute, when there's an abundance of water on one side of a membrane compared to the other side, it moves in or it moves out, depending on what direction, until it equalizes. And they show you this. It's a lab. We used to probably do this in high school. They used to have it was called an osmometer. It was ringing a bell. That type of stuff. You had like a little ball in there and you'd watch the level rise. And we do the same kind of a lab. But this is the reaction is really the important feature of this. And that's the distinction, again, to refresh your recollection, because any of you going into nursing, this is so critical. Isotonic media. Isotonic means it has the same relative concentration of solvent, whatever the solvents are, to solute of any living system that are there. So isotonic means it has the iso, the same tonicity, the same amount of water balance on as you have in your own system. And so what do when we infuse things intravenously? When we start an IV, okay, we use with extraordinarily rare exceptions, isotonic media. So these days, they start mostly with normal saline in our day, it was a lot of D5W, which is dextrose 5% in water. And I mean, it's based on, it's based on doing what's called stoichiometry. So you're able to look at the number of particles. Salt breaks up into two particles, sugar into one particle, the molecular weight, that whole thing. And you can do the math and figure out how many particles are there. So it doesn't matter. And then the other one that was used extensively is called lactated ringers. So if you think of sugar solution and salt solution, ringers is sort of like isotonic Gatorade, if you want to think of it that way. And a lot of, and it's very much a preference of the doctors that do that. But those are why those solutions are used, because we're not, unless we want to, we're not going to pull fluid into ourselves or push fluid out of ourselves. So if someone's dehydrated, we used to use occasionally use half normal saline. Okay. Because that would have, isotonically speaking, it would be somewhat hypotonic, push fluid, burn patients and things like that. Use a lot of lactated ringers in burn patients also. And so basically it's balanced. Net water movement is really not net water moving. Osmosis in the hypotonic, for us, it's devastating because we don't have a cell wall. So it'll cause the cell to burst or lyse. Whereas here, in bacterial cells with cell walls, it does not. It's just like plant cells. They're happy when you water them. And then now the nugget about hypertonicity, read that as salt or salt solutions or salty environments that are there. Remember, it's an envelope. So it pulls the envelope, the yellow membrane, away from that other color cell wall. And what will happen eventually, it will lyse. And in our cells, what happens, it forms something called crenation. So you get little, like bumps. It's no longer a circuit. It's no longer a uniform container. Might be the way to describe it. And then they go into passive and facilitated and carrier mediated. Not going to do it then. Whenever you all know, when you see the word active, when you see the word pump, it means that you have to expend ATP. Microorganisms don't like to expend a lot of ATP. It's not like us. We don't have a nervous system and a muscular system and a cardiac system where we have to expend a lot of ATP to do business. Okay. They don't have the resources. They don't do consistently aerobic respiration and make those wonderful, whatever it is, 36 to 38 ATPs per molecule of glucose. By and large, what you will see they do a lot of, they ferment. And believe me, you'll know that when you get into the micro lab, when you're cultivating cultures, because they have an aroma about them. That's because it's like when you open the refrigerator door and go, "Oh, it's fermentation." Whoa. So active transport, pumps, expenditure of energy, blah, 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 endocytosis. And again, microorganisms that we see other than the protozoan ones, because it's a membrane-based system, so it can't be bacteria or viruses. You'll see that there are some ways that our cells utilize endocytosis sometimes to bring them into the cell. That's a little bit different. This is on their end. So, I mean, we're the cellular form, we don't see any of these endo or phago or pino or exocytosis in bacteria because of the rigid cell wall. Who cares? And it's a photoautotroph, by the way. It's a plant. The temperature levels. The interesting thing about temperature, and I typically do this in AP1, so I'm sure you see it, because we talk about denaturing proteins. Remember, for those who have me, we go into, we talk about proteins and the whole primary, secondary, tertiary, quaternary structure. So most living systems utilize this. It's a bell curve. If you take everybody and you put them in and measure their height, you can have a middle ground. You have very tall people on one side, very small, whatever you want to do it. You want to do it by weight, you want to do it by anything. So typical bell curve, not drawn accurately, which means the optimal, the most common. In this case, when we're looking at growth, we might think of it this way, but this isn't the curve associated with temperature. Effectively, if we have a spectrum of values, usually the middle is where optimal activity occurs. And then and that's the whole way we scale tests if you want to do them fancy. And you had this, right? You had statistics, or you have some base background in statistics, standard deviations from the mean mode, all that type of stuff, right? Great curve. And we talk about it, and we'll talk about it in diseases as well. So it's a great curve. Not, it's not the curve. Okay. In temperature, it's a whole lot different. Temperature is not a bell curve. pH, when we get to, is a bell curve. Temperature is like this. The cold going to hot part of the curve as it's increasing, and let's say this is growth, being the y-axis, looks like a bell curve to start. But once it reaches its optimal temperature, the lethal temperature is very close to it. So T optimal, best, and T lethal are close together. Just like falling off of a cliff. When you and I get sick and our inflammatory process kicks in and we develop a fever, it's beneficial. It activates repair mechanisms, it activates our immune system up to a certain point. Our white blood cells are interacting with whatever the invader or the damage is. They are creating pyrogens many times, and those pyrogens are increasing that activity. And what happens? We get to an optimal, we go above our slightly optimal temperature. So we are just subtly increasing. But so we're at 37 degrees Celsius. That's our optimal temperature. Real normal, 98.6. Okay. If you increase this in degrees of Celsius by 3, so 3 degrees Celsius, 5.4 in the old Fahrenheit scale, we're going from 98.6 to 104. 104, you're going to go to the hospital most of the time. If you take it up another notch, now all of a sudden 2 more degrees, 5 degrees Celsius, 9 more, you're at 107. You're probably going to die. You'll get febrile convulsions, you'll have seizure disorders, etc. There'll be irreversible damage to the brain. You're probably going to die. Or you won't be very happy. Yeah. So that's the idea. And we utilize this in control of growth more than anything else, because what do we use to control? We cook things. We heat them. We boil them. And you're going to see what we do is we pressure cook it. That's the key issue, because that's how we sterilize the vast majority of things, plus other methods that we utilize a lot. That's why this is so important, because we realize that we can irreversibly denature the proteins by utilization of something that's relatively cheap and relatively effective, a lot less effective, and not really too harmful to the environment. If you want to think of it this way, the environment being us and surrounding people who are doing it. So there's a whole lot of nuances with regard to it. So that's the idea. Growth curve is a sudden drop off where and really the way you summarize it is the optimal temperature and the lethal temperature are typically close. And it applies to us, but it also applies to microorganisms. And organisms, typically microorganisms, there's about 5 ranges, 3 of which are significant to us. There are the extreme ranges, are probably not significant to us because most of us don't reside in those extreme environments. We're not in the middle of a volcanic caldera, and we're not somewhere in the polar ice pack. You may do so electively, but you see when doing research down in the South Pole, you can see all the accommodations they have to make to be able to do that. So lowest temperature is the minimum, okay. Maximum, highest temperature before proteins are denatured. So and again, lethal is very close to optimal, somewhere in between. So these are the categories we have. And every author has different categories. Psychro, not psycho. Psychro means very cold, extreme cold. Optimal temperatures are below significantly. Most of these are probably closer to freezing than that. So they will grow in freezing environments. Obligate, meaning when you see the term obligate, it means they can't grow above a certain thing. They're obligated to grow under certain conditions. They grow in refrigerators. I beg your pardon. It actually will increase because it's an increased degree of temperature. So when you're going to see psychrophiles are a little bit different. There's something that goes much better. These do not grow that well in the refrigerator. Okay, lakes, rivers, snowfields, polar ice cap, deep ocean, very, very cold. They typically are not pathogens. That's the first category. This is a significant category. Psychrotrophs. Troph means growth. So they grow in the cold. It's basically what occurs in your refrigerator. There's a certain degree of spoilage that occurs over time in a refrigerator. I mean, the most common spoilage agent that we see in dairy products, you're looking at two of them. Staphylococcus aureus, what I call public enemy number one when it comes to infections, is the SA of MRSA, and Listeria monocytogenes. And the point is, for you folks now, if you look online, every so look at the health stuff. Look at all the headlines. If you see one, there's an outbreak of this, or there's something of this, or 3 people died of this. Read about it. Okay. See what it is. You're going to see Listeria monocytogenes. Listeria is particularly nasty to people who are infirmed, who are sick, debilitated, or have what we now call comorbidities, and pregnant. Okay. So, because that's an unusual stressor for someone to have. And Listeria is very easy to acquire. It's typically Listeria is something that we find in animal feces that somehow, because things haven't been cleaned properly, and effectively it's a foodborne illness. And as you can see, it can flourish inside of a refrigerator, and that's a real worry. Every so I'm trying to think one of the more recent cases. It's like I do a whole routine about this. Is like it's like there was a case some years ago because think where they pick cantaloupes, melons off the ground, and when you pick stuff off the ground, if you didn't know that, because a lot of stuff gets picked off the ground, any food that's processed that goes to you undergoes what they call a power wash, wherever it's picked. Doesn't matter whether it's in this country or in Mexico, where a lot of our produce comes from. Think of the climate. Is it undergoes? You just hose it down with high velocity water to get rid of all the surface. If they don't do that effectively, okay, and they put it in a bag and ship it to your local, that's the whole green onion things when we talk about chi-chis and full-born illness. A very famous outbreak that happened here with hepatitis A. I first started teaching micro right around the time that happened. But Listeria, this one happened with cantaloupe. And so what a lot of people do, they go to the store, they grab a cantaloupe, looks great, feels right, shaking it, smells good, boom, take a knife, go through it. If you didn't wash the outside, what are you carrying into the inside? It's on the surface. So the edge of that knife is bringing it down. In surgery, we cut with a first knife, cut through the skin. We immediately discard that knife. We take it off the field and use a second knife to cut because you have skin contamination. No matter how well you prep the area, you can't get rid of it. That's how it works. So now you've done the interior, and we're eating what should ordinarily be a sterile interior. Moral of the story is scrub your fruit. Even if it says, you know how we love those bags of lettuce? It's easy to make a salad, pre-washed. Did you see them wash it? I didn't. I'm giving that sucker a bath. It's going right under there. Toss it around. I know it's easy. Just open it up, put it in there. Happy dance. Not this buckaroo. So, or there are organisms that are favor growth because refrigeration doesn't stop the growth; it slows it down. Okay, freezing is different. When we have most things that we do, and freezing is a whole different way of controlling things. So here are just some examples. So we do see organisms that and there's a lot of variants of normal organisms that actually favor cold weather. So, and I would say the range, let me just backtrack. They say 15 to 30. I would say maybe 20 to 40. So you see some of them actually can still grow even at room temperature or warmer temperature. So I'd say around 20. I'd say, maybe not. Maybe 15 to 30 is okay. This is the big category. This is the 20 to 40. And really, and these are mesophiles. This is the vast majority of things that impact us are mesophiles. That's why we refrigerate things, because we slow them from growing. And there's a lot of things that are stable in a refrigerator for a long period of time. Products with a substantial amount of fatty material like sour cream or cottage cheese, half and half, heavy cream. They last a long time. They have a long, look on the dates, they have a long freshness date. So some organisms, and we'll get into a lot of those things that are involved with regard to that, but these grow at temperatures roughly 20 to 40, maybe up to 45, depends on the author. Animals, plants, soil, water, temperature, tropical, subtropicals, temperate rather. That's us. Okay. And most human pathogens are, what makes them human pathogens? Their optimal temperature is right around our temperature. Some diseases favor, we'll get back to when we already heard a little bit about leprosy. Remember, I wrote about Mycobacterium leprae. Okay. Leprosy. Okay. Why did we see people with pieces of nose and toes and things falling off? It likes cold. It's a skosh below body temperature. So where are the areas we call acral parts on the tip where the circulations are worse. So there's a science behind that. Mesophiles are the big player, and mesophiles are where most of the things are. Our goal is to control them. We know we have certain things like the ones you saw before with psychotrophs. That has more to do with the product being processed not well or by mistake. And you should be aware of those things. I really do. I always look at those things in recalls. They're all published. Okay. It's the kind of thing, people, you have to pay a little more attention to that. Thermoduric, short exposures to high temperatures, but are normally mesophiles. Okay. This is where we see them because pasteurization is not sterilization, as you would see. And this is where the endospore formers come in. So that's why there's different methods of control when we have likely encounter endospores. Thermoduric, just think of it being durable with regard to temperature. Duro typically means strong, so strong with temperature. This is something that affects us. So that's really not a category. The categories you have to know are the psychotrophs, the mesophiles, and this bad boy, thermophiles. Optimally above 45 degrees, live in the soil and water associated with volcano. In other words, not in the middle of the volcano, but close to it. Compost piles, which a lot of people have. Direct sunlight, vary in range. Most forms of eukarya can't survive above that. That's pretty high. Okay. When you're getting to 60, now you're getting right around the level of pasteurization, about 140 degrees Fahrenheit. And then you have the extreme, what they call hyperthermophiles, which is another category you don't have to know. Those are the ones, volcanic caldera, that type of stuff, really where it's super hot, and they just don't live very well. Where do we see them? You could see them in hot springs. You could see them if you have a compost pile, which a lot of folks do at home. They compost. I don't see them so much around here. We do it. So we do that for my wife does organic gardening. So we have like adapted some big old one of those giant plastic containers and put holes in it, and you put stuff in it if you're a composter for soil, for the stuff that she's gardening. The other place you see it, and this is something you see more commonly, hot tubs. Hot tubs have their problems. Heated swimming pools, there's a lot of organisms. It's like when you scrape, and typically on a heated swimming pool, let's say that is typically like an in-ground pool, and you get scraped on. Then you have to be watchful about those types of things. Just saying. Because there are some organisms that will survive, and then and we'll get into the whole thing with pools and chlorination and chemical treatments, which is why they're a pain in the neck. We like them, but they're a pain in the neck. You saw that big pool we just came in where my daughter is, the ocean. I'm sure she'll. And this is the chart. So you can see falling off a cliff. Not quite so much cliff here. Mesophiles, big time cliff. Okay, that's the chart. So these are the ones you have to know. I beg your pardon. Psychotrophs, mesophiles, thermophiles. Oxygen levels. Very much different. I mean, to me, the one we have to deal with is oxygen. So oxygen, carbon dioxide oxygen has the greatest impact on it. It's because it's an oxidizing agent. It's the same problem we have with our cells. toxic derivatives of oxygen. Oxygen, when it's exposed to electrons that are free, and it makes those, and we went over this a little bit for those that had me for AEP. Remember what oxygen is? It's an electron acceptor. We bombard it with electrons, basically to create this O minus 2, so that we can join it with 2 protons and make water, which we can get rid of. And it's a safe product we use all the time, as opposed to oxygen by itself, periodically with oxygen like this, it'll get bombarded with electrons and it'll pick up an electron. So it'll be O2 with a minus sign. And the famous name for that is called superoxide. So all organisms, and it's a radical in the form of free radicals. And free radicals are the basis of DNA breakage. More free radicals and more DNA is likely to be compromised. Less free radicals, less DNA. Any oxygen utilizing organism has mechanisms to deal with these toxic derivatives of oxygen. That's the critical piece. In microlab, you will test for the absence or presence of one of these enzymes. It's got a very famous name called catalase. Organisms that can function in an aerobic environment have this, because what happens is the superoxide radical effectively is impacted by 2 enzymes to make it into water, okay, without balancing the equation chemically. The very famous enzyme, in addition to catalase, is called superoxide dismutase, SOD. So super and oxide, one word, dismutase, dismutase. Okay, so here's SOD. That converts the superoxide radical to hydrogen peroxide. You're going to find a very useful substance. It's also a free radical. So we only have a limited capacity to deal with that. The catalase rapidly changes hydrogen peroxide to water and oxygen gas. That's why when you scrape or cut yourself and you put some peroxide on it, what does it do? It bubbles. Your cells all contain catalase. It's been exposed to the hydrogen peroxide, it rapidly transforms it. That a large amount of oxygen gas also has another. Not only is it nice to have oxygen gas, bacteria don't like it because they have very limited capabilities. That's why it's an anti-infective. It creates toxic derivatives of oxygen in them. Love that. Hey, I love that. I like multitask. That's a multitasker. So the test that you do in the lab is by taking a culture, putting it on a slide, and adding a few drops of hydrogen peroxide and see if it bubbles. We call it the bubble test. It's the main way you differentiate between Staphylococcus and Streptococcus. Staphylococcus is aerobic. Streptococcus is not really. It's more in the anaerobic category. Most of its metabolism is anaerobic, and it doesn't have the catalase, which is the difference. So we move on. Okay, so microbes, those that use oxygen and detoxify it, that's the detoxification mechanism. Those that can neither use it nor detoxify it, those do not use it but can detoxify it. And you're going to see there's a whole category. You've got to think microorganism. Bacteria aren't like this. Okay. We all think about, well, we have aerobic metabolism. We have carbohydrates or other food substances. We effectively convert them to substances that we can chop down into molecules and effectively stick into, our mitochondria and generate ATP. And to do that, we require oxygen. So not all organisms, not bacteria, do this. Some bacteria only ferment. They don't have an electron transport chain. Okay, they don't have a proton motive force. Force is not moving. Actually, it's the Schwartz. Mel Brooks. Spaceballs, the movie. Do I have to show you the video? The poster on my door in my office. You come to see me. Spaceballs the movie. Spaceballs the amusement park. Spaceballs the lunchbox. Intercellular reactions, it's transformed into several toxic products. So here are some of them. Singlet oxygen, very reactive, not worry about it. Superoxide, worse. Hydrogen peroxide, the hydroxyl radical is another one. So when you have these, effectively what they do is they bump into stable molecules and destabilize them and create other free radicals. That's really what happens. A free radical is the linchpin of modern cancer theory. So these are all what we call oxidants. And that's why you and I, when you hear about superfoods or certain foods and certain diets or vitamins, they go rich in antioxidants. That's the reason you're trying to supplement your own superoxide dismutase and catalase in such a way that you have an extra abundance of antioxidants. And some people erroneously think that it's not going to protect them if they do something that's going to expose them to too much. It's not a be all and end all. This is them balancing the equation. Don't want to do that. So let's look at the categories. So there's a classic. I'd love to have them do this in lab, and I've done it. It's not so easy to do because you need the right kind of microbes to do it. Basically, what you do is, and you may have, did you start to inoculate tubes of broth yet? Okay. Effectively, what you do is you heat a tube of broth, a particular broth, and you inoculate it with, you use the loop, flame the loop. Okay. You flame the loop, you obtain a piece of the organism, you put it in there, kind of use the loop as a stirrer, seal it. Obviously, we sterilize the loop. Okay. So you do that with different organisms, and what you're able to see based on where they grow. As you might guess, things that are anaerobic grow at the bottom of the tube. Things that are aerobic, top of the tube. Things don't care, grow all over the tube. So we're able to categorize them based on this. So it's a classic experiment. It works pretty well. And again, I think you'd have to have specific lab things to do it. And so we'll comment on that. Aerobes. So when we talk about obligate aerobe, it has to have, we are obligate aerobes. We die without oxygen. Can use oxygen in their metabolism. The enzymes you see over there, superoxide dismutase and catalase, to detoxify oxygen products. Organisms that cannot grow without it, like us, obligate aerobes. So we're in that category. Most fungi, protozoa, many bacteria, obligate aerobes. And if you're thinking about us, aerobes. So you can see where the growth is in the top. Some organisms are harmed by normal atmospheric. They don't have enough of these agents to prevent toxic derivatives from damaging. What is less than normal? Normal oxygen concentration. Everybody knows this. We're an 80-20 planet, 80% nitrogen, 20% oxygen. We say in the land of Star Trek, Class M. Live long and prosper. For those who didn't have me before, my son, now the commander of the destroyer, and I used to go to Star Trek conventions in costume, which I'm sure you'd all believe. Don't like Star Trek? Okay. I give up. Nevertheless, we have an 80-20 planet. 2 to 10% oxygen. They can only tolerate about as much as half the oxygen that we have. So roughly it's 2 to 10%. So they are harmed by normal, require a small amount for metabolism. So there's different ones. These are monstrously important. Okay. Helicobacter pylori. A lot of people think, I do not, that it is, if they tell you it is responsible for gastric ulcers. It is not. Is it associated with gastric ulcers? It is. Is it more likely as a cause of gastric cancer? Yes, probably. I'd buy that. Helicobacter pylori has a flagella, and what it does is it burrows into the gut, into the stomach lining, just at a level so it has less oxygen, and then it flourishes and it damages that lining, and that creates ulcerations. So I'd say 90% of the people who have gastric ulcers have this. You do a when you do an endoscopy, you take a swab. If you're familiar with that, you put a fiber optic tube down. I've had that done 15 times. They've never found it. And I've had two monstrous surgeries for gastric ulcers. I used to get ulcers, now I give them. Better to give than receive, right? It's very Christmassy. My wife still has her Christmas tree up. The dog's very happy. Decorative purposes only, Lincoln. Yes, ma'am.
Why is the H. pylori test in the breathing tube, like when you exhale into it? Why is that?
I don't know. Short answer. It's probably because it's got. I don't know.
Like
But
something.
it's probably something. There's enough of it in, that comes out when you're breathing. That does that. H. pylori is a very interesting bacteria in that regard. So that's one of the ones. That's a microaerophile. Anybody know what Borrelia burgdorferi is? Oh, very famous disease. It's the most common vector-borne disease in the United States. Most common spread by, in this case, let's say insect. I live in an area where it's constantly there, as do you. Anybody who lives around here. Lyme disease, correct. Because it's associated with it. This vector is the deer tick, sometimes the wood tick. And we live in Deer Central. I walk the dog, there's deer. Borrelia burgdorferi. Okay, just throwing it out there. It's not the only one. There's a strain of Streptococcus, okay, Strep pyogenes, which we'll learn about, and we go over all those conditions. Strep pyogenes, which is the strep throat strep, and it's also a skin infection strep that causes the flesh-eating disease. There's a number of bacteria that do it. Most commonly, it's Streptococcus. It's a microaerophilic Streptococcus. It's got a fancy name. It's called necrotizing. When necrosis is something dying, necrotizing means it's almost dying and being liquefied. So necrotizing fasciitis, your body is divided by what are called fascial planes. So they're the internal dividers between skin and fat and muscle, ligaments, tendons. It dissolves those. So basically, neck fash, which is the name we give it, it's caused. What we see is the skin stays intact and the bone stays intact, and everything in between just almost liquefies and has tons and tons of gas because it's a gas producer. I've seen 2 cases. And I saw 2 in 20 plus years of practice. So it's not that common. But you hear about it all the time because they do that. It's interesting. I don't know if it's the same organism. There was a real uptick in those classes when they had all that flooding from the hurricane in Houston about 5 years ago or so. Those are microaerophiles. So we've got 2 categories: obligate aerobes, microaerobes. Let me see how we're doing. Oh, we got plenty of time. Facultative aerobes. Facultative, 2 important words. Facultative, when you're in the lab, you're using facultative organisms. They grow easily. That's why we get them, so you guys can study them. So facultative means grows easily. Doesn't need a lot of TLC. Okay. The opposite of that is fastidious. Fastidious organisms require growth factor. Streptococcus is a fastidious organism. That's why it kind of hangs out in our throat. There's a lot of nutrients. It's tough to grow. In general, things that grow easily are hard to kill because they grow easily. Staph aureus, E. coli. Things that are tough to grow are easy to kill. Streptococcal infections are really easy to get rid of. A lot of antibiotics. Syphilis, one of the most famous diseases of all time. You can't even grow it in the lab. It's so hard to grow. You can't grow it in culture. Very easy to kill. Not too easy to stop the spread. That's a whole different story because it's sexually transmitted disease. The E. coli you're using in the lab when you start to use it. All those organisms, they're all, nearly all of them, not Staph aureus or Staph epidermidis, facultative anaerobes. they're technically aerobes because they can function in an oxygen environment. That's why we can grow them in the lab. They do not require oxygen for metabolism, but use it when it's present. So they can flip flop, if you will, between aerobes and anaerobes. They're facultative. Okay, they have that ability. Many gram-negative intestinal bacteria and Staphylococcus, and gram-negative bacteria are so common infectious problems. Urinary tract, because from fecal contamination, urinary tract problems, skin problems, people who are incontinent. When you get to the hospital, you'll be sick and tired of this. Absolute or obligate anaerobes. So we've got three categories. Obligate anaerobes. They can't function in oxygen. We kill them with oxygen. The aforementioned hydrogen peroxide is great because it forces oxygen into the area. They don't have their anaerobic organisms. They do have all of these ones we've looked at so far. They do have electron transport chain. They do make ATP by a way that's similar to ours. These do as well, except they do not require oxygen. Because oxygen's job is called the terminal electron acceptor. It sucks up electrons. You got to have a parking place for electrons when you do metabolism of this type. They use sulfur and nitrogen, so they don't make H2O. They make H2S, a lot of them. Hydrogen sulfide, rotten egg smell. So smell test is important for anaerobic infections. Gangrene, someone has gangrene. C. diff. Those are anaerobic infections. You can make the diagnosis outside the room. Science. It's a good thing. Lack the metabolic enzymes for using oxygen and respiration. They use other ones. So they also lack the enzymes. They don't have SOD. They don't have catalase. Oral bacteria, streptococci are a lot of those. Intestinal bacteria, many are like that as well. That's where the C. diff comes in. All that Clostridia family. Tetanus, botulism, they're all that. So they grow at the bottom of the tube. So if you look at the tube so far, facultative throughout the tube. Aerobes at the top, obligate, microaerophiles down from the top because they don't want all that oxygen in their way. Facultative throughout. Anaerobes, bottom of the tube. Now, the last ones, the interesting ones. Aerotolerant anaerobes. Do not utilize oxygen but can survive and grow to a limited extent because they're not harmed by oxygen mainly because they possess alternate mechanisms for breaking down peroxides and superoxides. So they have other ways of dealing with oxygen before it becomes toxic. I beg your pardon. So another name we give them is obligate fermenters. They still have to make energy. They don't have an electron transport or similar mechanism as all the others do. How do those facultative anaerobes that we saw before? They ferment. So fermenting is a big, big deal. Lactobacillus, certain kinds of streptococci, clostridial species. That's why they harm us a little bit more, even when they're out in the oxygen, because they're not true obligate anaerobes that are there. And so what you will see with regard to these aerotolerants, they're big players. Lactobacillus is the single most important bacillus in dealing with sexually transmitted diseases in women. Lactobacillus are very easy to kill bacteria. So broad antibiotics clobber it. What it does is it ferments glycogen. Estrogen in women of childbearing age produces scads of glycogen in the female reproductive area, vaginally, urinary tract wise. And what that does by fermenting that glycogen, it produces a ton of acid. Acid's the barrier to infection. In prepubescent girls and in menopausal women, postmenopausal women, now we rely on mucus as a barrier. It's not nearly as effective. So we revisit this again when we get to urinary tract infections. It's a ton of it. I'm better at A&P, but I like teaching micro. Better. Capnophiles, higher CO2 tension. This is the problem with isolating these. Okay, strep pneumonia functions. Most of the time we see it. It's the leading cause of bacterial pneumonia. Neisseria, gonorrhea, another one, very, very hard to cultivate. Meningitis, the leading cause in adults of bacterial meningitis. Historically, one of the most difficult sexually transmitted diseases. Brucella, which is a particular fever. You don't see a lot of that. Brucellosis has to. I'm trying to think of where it comes from, where the bacteria. I'll get back to you. So that's all there. And it's pH. There's nothing to really say about it. Okay, we know with pH 7.0 is almost any organism that we have is going to grow roughly between 6 and 8. Okay, we call them neutrophiles. There's a white blood cell called a neutrophil. They're spelled the same except the file has an E at the end. So most of the bacteria that we deal with, the alkaphiles and acidophiles are we just don't see a lot of them. Okay, there's some odd bacteria that are there. Proteus can create those conditions. We do deal with Proteus, but Proteus can still grow fairly easily in pH environments. So they don't have the term there, and they should, unless I missed it. We call them neutrophiles. I think that's the one to remember. We'll stop here. We'll start with osmotics and halophiles, and that'll bring us into the growth curve. So we'll get done with this chapter on Monday. Thanks. And we're done.